Everyone, welcome back to the second part or the second subtopic of the introduction to human brain, which is the brain mapping techniques. Okay, so moving on real quick to brain mapping. So what is brain mapping? As you can see from the pictures at the left side of the slide, there is the brain on top of a globe or a map. So brain mapping here is the attempt to provide a complete picture about how the brain works. We wanted to have a three-dimensional map of the brain representing the surface, structures within, and the function performed by components of structures. Meaning we wanted to know where is the part on the brain function as something. Alright? So brain mapping is to provide a complete picture of how the brain works. And then in this subtopic, we will cover two different brain mapping techniques, which are structural and also functional. So for structural brain mapping techniques, you will be exploring the static images of brain structures. But for functional, you will explore the dynamic images of brain activity. Right? So the focus of structural brain mapping techniques here is to examine the brain anatomy and morphology, while for functional, we wanted to study the brain activity and function. So one you will see a very static images of brain, and one you will see a very dynamic images of brain activity, right? So for structural, we have two brain mapping techniques, which are X-ray techniques and magnetic resonance imaging, the MRI techniques. Okay, and for functional, we have also two electrophysiological techniques and blood flow techniques. So let's go to the structural brain mapping techniques first. Okay, so we'll cover the X-ray techniques first. Okay, remember this is the kind of pictures to reflect the X-ray. Okay, the pictures at the left bottom of the slides. Okay, so first one, we have cerebral angiography. Cerebral angiography, useful in detecting disorders associated with abnormalities in brain arteries. For example, when someone have a hemorrhage, a heavy bleeding, or loss of oxygen in that area. Meaning that for this cerebral angiography, we wanted to detect abnormalities in brain arteries. Okay, in the brain arteries. And this procedure uses a special dye, an X-ray, to see how blood flows through the brain. Because it is because the cerebral angiography is part of the X-ray techniques, then there will be some radiation, okay, involved, because there is X-ray and a special dye. This special dye will detect the brain arteries later on. Okay. And then for the second one, we have computed tomography or CT scan. Computed tomography or CT scan. So computed tomography meaning it is a representation of a section. Okay, because tomograph itself means image sliced through an organ without the interference of all layers of tissue present in X-ray images. The X-ray films appear blurry because they are 2D rendition of our 3D body. So CT scan, okay, as you can imagine, this is how CT scan brain image looks like. Okay, it's very blurry. Okay, you can't really tell what parts of the brain it is. Okay, because it's not very clear. Next is the computed axial tomography, the CAT, developed in 1970s. And it is almost similar to the CT scan, but have less radiation when compared to the traditional X-ray. Less radiation here means that it still has the radiation effect, but in less quantity, okay, in small quantity. Because this computed axial tomography, the CAT scan technique, is still an X-ray technique. So when it is an X-ray, there is still radiation going on. However, with the computer technology, they enable deep blurring of picture and reconstruction of a complete brain image from multiple views. And this CAT scan rotates works with a rotation of fan-shaped X-ray beam around a single cross-section. And this development of CAT enabled process of imaging brain internal structure to become safer and more precise. Why safer? Because less radiation. Why more precise? Because there is de-blurring effect from the computer technology. So just like how I explained just now, for the advantages, it's more safer, it is more precise, and it have clearer image. Alright, but it doesn't indicate when an activity is occurring in the brain. Because why? Because this is a structural brain mapping techniques. You will only be presented with a static brain image picture, right? And last but not least, for structural brain mapping techniques, we have the magnetic resonance imaging, the MRI. I think most of us are familiar with this word MRI, but maybe we have no deeper knowledge about what MRI really is. So MRI is a magnetic resonance imaging, a new method developed in 1980s. Okay, remember just now the CAT scan? This CAT scan was in 1970s, and a decade later we have MRI. And as the name suggested, it uses a magnetic field to take images of the inside of your body. So assume atoms in the body will react to the magnetic field, okay, based on radio signals emitted by the protons in the human body. So in our body there is radio signals, okay? Sorry, there is protons, okay, in our blood, in our water contained in our body, and these will react to the magnetic field of the MRI. So the advantages of using MRI is there is no X-ray or radioactive material use. Okay, as you can see here, X-ray technique only covers cerebral angiography, computed tomography, computer axial tomography, CA, CT, and CAT. But magnetic resonance imaging itself is using the magnetic. Okay, the MRI itself is not part of the X-ray, meaning when it is not part of X-ray, it doesn't have radioactive material. The resolution of the image is sharper when compared to the CAT scans, and the flexibility allows the researcher to distinguish different structure of the brain, the gray or white matter and cerebral spinal fluid. However, the disadvantages of this technique is it is very, very slow for an image to be generated. Therefore, it might no longer depict a situation precisely because it was at the time of the imaging only, meaning that you cannot expect to have a brain image of the current time, okay? Because this is a very slow process, meaning that the image generated maybe from 2 days or 3 days ago, okay? And for MRI, we cannot be used in patients with metallic devices like pacemaker. So anything metallic, anything that can be attracted to magnet cannot be worn during this procedure. Okay, why? Later we'll discover. And then this technique also may cause claustrophobic. So what is claustrophobic? Claustrophobic is the fear of getting trapped in a very small and cramped space. Alright? So again, just now what I asked, what would happen if you wear anything metallic during an MRI? This is what happened. Okay? Anything that a magnet can attract, anything metallic, will be sucked into the MRI. So do not wear or do not patient with any metallic devices in their body should not use this procedure, right? Because it can be very dangerous. Alright, moving to the next brain mapping techniques, which is the functional brain mapping techniques, involving the electrophysiological technique and blood flow techniques. So for functional brain mapping techniques, just now we will see the brain activity or the dynamic brain image. Alright, the dynamic images of brain activity. We wanted to see the changes in brain activity, meaning there might be a video, there might be a graph showing how our brain activities changes over different cognitive tasks, right? So the first one we'll cover from the electrophysiological techniques is the electroencephalography, the EEGs. Okay, it is a taste of the function of the brain itself that records the electrical activity on the brain surface and image the brain while it is performing cognitive tasks. So these techniques detect the location and magnitude of brain activity involved in the various types of cognitive function. It's very non-invasive and painless procedure because they will the person will only apply the electrode leads to the patient's scalp. So we don't have to go and lie and go through the MRI scanner or the CT scanner, but this EEG will then worn by the patient on their scalp, and the electrical activity will be recorded. Okay, the electrical activity on the brain will be recorded. So electrophysiological techniques, as you can see, is about is about electrical activity on the brain. While physiological, we can remember how this technique is done. So physio mean the body, okay, the anatomy, so the part of our body. So this technique, electrodes will be placed on their head, on their scalp, to test the electrical activity on the brain surface. Okay, but for the EEGs, we do not. Okay, for the electrophysiological techniques, we will not be presented with any brain images, but we will see the graph, the electrical activity, a brain activity graph, right? Just like here, we will wear the EEG cap that will be connected to the electrode and amplified by the amplifier to measure the potential for each electrode. So the tracing and recordings of patterns of electrical activity in the brain, changes in brain activity during performance of cognitive tasks, and then provide fine-tuned reading. So as you can see, here is the result of the EEG. This is our electrical activity when we are excited, when we relax, when we feel drowsy, when we asleep, and when we are in deep sleep. Okay, so it changes over different cognitive tasks, right? So again, the advantages of EEG is we can trace rapid changes in neural activity, and we record activation of people who are fully conscious and engaged in various activities. However, for the disadvantages, invasiveness relative may affect data validity, and there are possible interference from movement, heartbeats, and blur reading. Why? Because these people, okay, are fully conscious and they are engaged in various activities. So they might not, they might zone out. They might not give their full focus on the instructions given. So other things like movement, heartbeats, maybe they were zoning out, might be possible interference that affect the data validity. So another part of the electrophysiological techniques is the event-related potential, the ERP. Okay, what is event-related potential? As the name suggested, subject will be presented with a stimulus or an event during EEG process, and voltage change around the stimulus before and after will be recorded. Okay, to find average values and evoke response, provide a picture of neural activity changing over time as the brain processes information. Okay, we wanted to see the voltage change around the present of stimulus before and after the present of the stimulus, right? And we wanted to see the average values. Okay, so as the name suggested, event-related potential is when the subject is presented with stimulus or event. Okay, and then we move on to the blood flow techniques. Okay, no longer about electrical activity, brain activity. We're no longer looking at the graph, but now we are back to looking at the brain images. Okay, so blood flow techniques, the first one is positron emission tomography, the PET scan. This is one of the most famous brain mapping or brain imaging techniques, right? So put your focus more on these slides. Okay, so what is PET scan? PET scan is a method of measuring cerebral blood flow while subject carries out cognitive tasks. Okay, uses radioactive isotopes. Okay, for example, positrons, or people also call it as a glucose solution containing short-lived radioactive tracer. So PET scans look at bodily process by detecting the decay product from radioactive tracers injected into the body. Alright, so the radioactive tracers will be injected into the body, and then they will go through this PET scanner, and then this is the kind of pictures, the kind of brain images diagram that it will give you. A very vibrant color, you know, where the blood flow in brain shifts to different locations depending on tasks being performed. Alright? The advantages of PET scan here is it provide an image of brain activity as a cognitive task is occurring. Okay? We do something. While we do something, we can see the part of the brain that is active. As you can see from the picture down here, the occipital lobe lights up or is active when the person is passively viewing words. Viewing, okay? They see the words. So when something to do with vision, the occipital lobe will be activated. Just like here, listening to words, the temporal lobes will be activated. However, the disadvantages of PET scan just now is it requires the use of cyclotron, expensive equipment to provide the radioactive isotopes, and not as sensitive to changes over milliseconds, not as precise when compared to the electrophysiological techniques. And just now, when we talk about positron emission tomography, we will be injected with radioactive material. When we injected something radioactive in our body, then there will be radiation. So this is less safe. Okay, so that is the drawback of using PET scan. Then we have single photon emission computed tomography, the SPECT. Okay, the SPECT scanner, unlike the magnetic resonance imaging, the MRI, we don't use magnet. Okay, we don't use the magnetic feature, but we use the gamma camera and the gamma rays. Okay, it is a type of nuclear medicine test that uses a radio tracer that is injected through your vein. This one also need you to be injected with something. This one is a radio tracer, where it shows how blood flows to tissues and organs and creates a detailed 3D map of the blood flow activity in your brain. So as you can see from this picture down here, this is the kind of picture SPECT will give you. Okay? The brain image that SPECT will give you. Very colorful, very 3D, very fancy looking brain, you know. However, you still need to be injected with radio tracer because this is a nuclear medicine test, right? And last but not least, we have the functional magnetic resonance, the fMRI. Okay, just now we have MRI, right, in the structural brain mapping techniques, but in functional brain mapping techniques, just like the name, we have functional magnetic resonance, the fMRI. So the functional magnetic resonance, also known as real-time or dynamic MRI. Functional equals to real-time equals to dynamic MRI. Available in 1990s. Okay, let's recap back. In 1970s, we have the CAT scan, CAT, the CAT scan, and then 10 years later, 1980s, we have the MRI scan, and then another 10 years later, in 1990s, we have the fMRI. That's why it was called the decade of the brain. And this technique is used to detect changes in magnetic state of blood using MRI scanners with fast imaging techniques, record changes in oxygen level, blood flow in various brain locations when subject perform various cognitive tasks. So fMRI is a technique for determining which part of the brain are activated by different type of physical sensation or activity. Okay, so the difference between fMRI and the MRI is that the functional one, we see how the brain activity. we see how dynamic the brain images can be with our brain activity, while for MRI we only see the static brain image only. The advantages of using fMRI is it provides information about brain structure and function, of course, and it is safer and less harm to patient as no X-rays or radioactive material is used. Okay, we do not inject anything into our body, or we do not use X-ray during these procedures, so it is safer, and it allows researchers to infer which locations are involved in specific activities. So these are the summary, okay, of this subtopic, where you can see this is the picture of a brain image that CT scan give you. This is the difference between the MRI. You can see it more clearly. In the PET scan, you see a very colorful one. Okay, the PET scan here, very colorful one, and for fMRI, you can see more clear version of MRI. So you can see the activated part of the brain activity, and for EEG, you will see this graph, electrical brain activity graph. So summary for both subtopics here. In the brain structures and functions, in the first part of the pre-recorded lecture video that you have watched just now, we have covered the human brain, consists of brainstem, cerebellum, and cerebrum, and cerebral cortex consists of 4 components: frontal, parietal, occipital, and temporal, where you can use the mnemonic that I provided you with, which is the fancy penguin occasionally twerk. Alright, so next we have the corpus callosum that connects the 2 hemispheres, and we also have the nervous system divided into 2, the CNS and the PNS. Okay, the central nervous system. Okay, CNS is a brain and spinal cord. Peripheral, we have the afferent and efferent, senses to the brain, brain to the senses, and neurons, we have the dendrites, soma, axon. Sorry, here is synapse and neurotransmitter. Okay, that was what we have covered in the first subtopic, which is the brain structures and function. Or for the brain mapping techniques, we have covered the structural and functional techniques involving X-ray techniques, magnetic resonance imaging and MRI, electrophysiological techniques, and blood flow techniques. So I think that's all for today. Thank you for bearing with me, and hopefully you have a wonderful day. Bye bye.